All right. Well, thank you everyone for joining us. I'm super excited for this week's Science Cafe. Dr. Brendan Harley will present his work. I'm super excited to see how this goes. So take it away.
Thank you. Welcome, everyone. So as a quick introduction, my name is Brendan Harley. I'm a professor of chemical and biomolecular engineering here at the University of Illinois. And I have an appointment at the Institute for Genomic Biology as well. I'm the theme leader of the regenerative biology and tissue engineering theme. And so it's a real treat to talk about some of the work we're doing in my lab and talk about some of the ways in which it impacts healthcare. So I'm going to talk about this idea of what building tissues mean. And so let's get rolling. I think one of the things that I like to start off with is talk about what it is that we do in my lab. We do really cool science, but I think one of the things that we try to do as a group is think about ways that we can make an impact. And the mission that we have is a little bit broader. We're interested largely in developing biomaterials, and I'm hopeful by the end of this period, we'll talk a little bit about what biomaterials mean to my group and what we do. But really the goal we want to think about is ways that we can establish an inclusive and supportive research environment. So I'm going to talk about the way in which some of our motivation is driven by the trainees in my group going off in really exciting directions, and the ways that some of our research is driven by our collaborators that we're working with. But our goal in the lab is to develop biomaterials that mimic what we call about complex tissues. And I'm going to talk a little about what that means. But really our idea is to develop materials that can be implanted in the body, and this to induce regeneration. So as opposed to like an artificial hip, that's a permanent piece of metal and plastic that gets implanted, we're thinking about materials that could be implanted as a scaffolding that over time will be replaced by your real tissue, and thinking about ways to drive regeneration. We also think about developing materials as models of tissues we can use outside of the body in the laboratory to study processes associated with disease progression or to validate new therapies. And I'll talk about a little bit about that as well. But the thing that really motivates us is that if you look inside the tissues and the organs of our body, they're not a uniform sack of cells sitting in an amorphous structure. Our tissues and organs are incredibly complex. And the things that inspire my group are the interfaces, transitions, and heterogeneity associated with tissues. So some examples. Lots of tissues in our body are spatially graded, like the insertion between the rotator cuff and the shoulder is a tissue where you go from muscle to tendon to a fibrocartilage enthesis to bone. And those transitions are incredibly important spatially to enable function, the ability to raise your hand. Lots of tissues change in time with development. The image here on the bottom is meant to represent osteoporosis, the loss of bone mass you see particularly in women, but it's a transition that happens over time. So a lot of these tissue environments change in time. Most of the tissues in our body have more than one cell type in them. There's a huge degree of cellular heterogeneity. So you have stem cells and mature cells, vascular cells, neural cells. They're all working in concert and in a coordinated fashion. And lastly, most of these tissues, in addition to having spatial and temporal gradients and being highly heterogeneous in terms of cells, they have structure associated with them. They may be hierarchical in structure, like in the kidneys and livers in terms of how the tissue organization drives function. Or they may be multi-scale. They have properties at different levels, such as bone, which will have a mechanical property at the scale of your femur. But if you look inside of the femur, it's organized into trabeculae on the micron scale that still have their own mechanical properties associated with them. So these degrees of heterogeneity are everywhere in our body, and they're really hard to replicate. We don't have ways in which we can perfectly replicate any of these tissues in our body yet. And so we're thinking about strategies to develop materials to let us replicate features of these tissue environments and use that as a starting point. And that's really the thing that motivates a lot of our work. And I'm going to show you a couple of examples, one related to regenerative medicine and one related to a model of cancer progression that we use, where we're trying to replicate not the entire tissue in its entirety, but key drivers to enable what we want to have happen. One other question I just want to bring up to think about is that when we start thinking about developing biomaterials, one of the questions that we run into a lot is, what can we do to go beyond the idea of generalization? So what do I mean by that? One strategy that has been pursued in the past is, for example, I'm going to talk about bone regeneration in a few minutes. And we might think about developing a single biomaterial implant we could use for lots of different patients that are facing a critical size bone defect, and we're trying to regenerate that bone. The reality, though, is there's lots of variations between us that we don't do a really good job of understanding yet in terms of age, ethnicity, underlying health conditions such as diabetes. All of these things can drive differences in how we heal after an injury. And so it's important for us to not just think about creating a one-stop material that solves everyone's problems, but think about some of the contextual complexity associated with healthcare and ways we can address that. And I'm going to talk about one example where we're reimagining the use of our biomaterials to ask questions about patient variation. There's also ways in which we're trying to do this to think about new challenges using our materials as lenses to study new types of problems that I won't be able to get into today, but I think I'll at least illustrate this idea a little bit, that we're trying to think at the IGB about ways that we can address all members of our community with some of these solutions. All right, so I'm going to talk at first about some examples in musculoskeletal tissue engineering. This idea that we are creating biomaterials to implant in the body to induce healing. And I'm going to talk primarily about an example of craniofacial bone regeneration. But I think it's also important to just note that this idea of tissue engineering, that you take a material and add a cell and some other growth factors or cytokines and use that to regenerate tissues, has been around for a couple of decades. And there's not a lot of clinical solutions that are out there yet. And I think that's something that our community is starting to really grapple with. And so a lot of the state-of-the-art methods to heal craniofacial bone defects, like the ones we work on still, are largely permanent implants and non-regenerative prostheses. And we're working very hard to identify the challenges preventing us from making a clinically relevant solution. And I'm going to talk about two of them today in the spirit of how do we develop a material that can be implantable and can induce regeneration of large bone defects and do so in a manner that is surgically practical. And so just as a quick warning, we have an image on the next slide that, while it's not graphic, is heartbreaking all the same. And so one of the major challenges that we have in our field is we're thinking about challenges for craniofacial bone repair. So these craniofacial bone injuries involve significant amounts of trauma and are really common in our society. We have congenital defects such as cleft palate. We have post-oncologic defects. So we have a salivary gland cancer where a patient may have a large portion of their jaw removed, during the cancer removal. We also have lots of different high energy impacts for sports injuries, car accidents. We got into this work funded by the US Army. They were very interested in survivable battlefield injuries being experienced by warfighters. More than a quarter of all of them experienced in Iraq and Afghanistan were penetrating craniofacial injuries. And when you think about it, it makes sense. We've made massive improvements over the last few decades in body armor and vehicle armor. And one of the few areas on our warfighters that isn't properly armored is the face. So you see disproportionate amounts of massive injuries, such as this IED injury you see on a scan on the left. And these injuries are large, they're traumatic. And the goal that we have is to develop a biomaterial that can be shaped quickly and precisely in the operating room to custom fit each defect. Each patient is going to have a unique implant. And so it's not plausible for us to think about creating a one-off implant every time, but we're thinking about how do we develop a material that can be cut and shaped rapidly by the surgeon intraoperatively, like they typically do right now with non-regenerative implants, but they would act in a regenerative fashion to grow back new tissue. And that's really the clinical goal we're pursuing. So there's a lot of challenges associated with this work. You think about right after injury, we want to think about our ability to create an implant that is customizable, will conformally fit into a unique defect for each patient, is strong enough to sit in a bone injury model, and will allow cells to grow into it in order to aid healing. Over days to weeks after injury, we're thinking a lot about the immune response, where you have macrophages and other cells are responsible for cleaning up the wound site. So we're thinking about ways that we not necessarily get rid of this immune response, but how do we shape and mold this immune response to maximally support new tissue growth? And then we want to have systems that we can assess how well we can do. Large scale defects where we can ask questions over weeks to months. How well can we regenerate tissue in clinically relevant defect models? So I'm going to talk about some of the work we're doing in that space. Obviously, in this type of a talk, I'm not going to go into super detail, but I want to give you a flavor for how we go about doing this work, and I'd be happy to answer questions after we're done. So one of the major questions we've worked on in my lab is how do we develop a material to support this type of work. And so we worked for a very long time developing a class of collagen biomaterial known as the collagen scaffold. These are materials that look very much like kitchen sponges. They're dry, they're porous, they can be hydrated and have cells grow into them. And you see an image here on the left of what one of these looks like if you zoom in really close. The scale bar here on the bottom is about 100 microns, about the size of a human hair. So what we're seeing here is a porous network where the lighter regions are the collagen fibers and the darker regions are pores. This material is more than 95% pores, which means it's very easy for cells to move into these large materials. It's easy for nutrients and oxygen to diffuse through these materials. The way we make these is really interesting. We start with a liquid suspension of collagen, which is one of the native proteins in the tissues and organs of our body. We take collagen and other proteoglycans and we mix them in an aqueous suspension to form a solution that looks kind of like milk. And then we freeze it. And as you freeze it, you form ice crystals in the aqueous phase. And so by controlling how fast you freeze or how cold you freeze the material, you can control the size and the shape of the ice crystals that form. After it's frozen, we then sublimate this material. So that means we drop the pressure and increase the temperature, and you can go directly from solid to vapor. So we pull out all the ice crystals as vapor, and what we're left behind is a porous network whose pores are defined by the size and the shape of the ice crystals we form. So we did a lot of work over the last 20 years to develop processing conditions by which we can create really spherical and uniform pores, ellipsoidal and aligned pores to make anisotropic structures, and to add in other ingredients such as mineral nanocrystallites to mimic bone. You know, bone that we see in our body has a lot of collagen, has a bunch of mineral in it. So we tried to develop porous scaffolds that would have the same composition as the bones in our body. And we have a variant that we've been very successful with over the last few years. So this material it's about 40% weight mineral. It's got a calcium phosphate mineral like we have in our bones. We've tried higher and lower amounts of calcium phosphate. This is sort of inspired by development where our bones start out not being quite as mineralized as they are in adulthood. This seems to work really well from a healing standpoint. This mineralized scaffold is really exciting to us. And one of the reasons is that we can add in stem cells we get from patients taken out of the bone marrow or fat through a liposuction, where we can isolate stem cells that have the ability to turn into bone cells. And so one of the major challenges in our field is how do you drive this efficiently? If you want to treat a patient, you want to get stem cells from the patient, you want to put it into the material, and you want to have those stem cells, which can proliferate a lot, turn into lots of bone cells. The traditional way one does it is you add a lot of growth factor, particularly something known as BMP2. It's a cytokine that will drive differentiation of these stem cells into bone. It is used clinically, but some of the challenges are it's incredibly expensive and it's very hard to keep this small molecule just in your material. And so you end up using a lot of growth factor and it can diffuse outside of the wound site and form little bone nodules away from where it's supposed to. So this material we designed in a number of ways, but one of the primary ones, and we see on the bottom here, these are mineralized materials that their structure and their chemistry is such that it instructs the stem cells inside of them to turn into bone cells without us having to use that growth factor. We can see that on the right where these non-mineralized scaffolds, you see very little red, which is a marker for new bone formation in the laboratory. You can add in, the scaffold on its own, you can add in cells and this BMP2 growth factor, you see a little bit of bone formation. Whereas on the bottom, our mineralized scaffold, you added cells just on their own and you get lots of bone formation. We can add in the growth factor and we see the same amount. We then went into animal models. And so this is a model from a rabbit where we can take on non-mineralized scaffolds and we see largely just soft tissue that forms in the defect. So this is a defect in the skull of a rabbit. It's known as a critical size defect, meaning if we do nothing to it, it won't heal. And you see largely these non-mineralized scaffolds on the top, like over here on the left. You see very little new bone formation in these blue regions in the middle, which is where the hole was. However, on our mineralized scaffolds, we see really efficient bone formation. And so the fact that we can take stem cells, put them in our material and have the material itself drive the process we want means that in a matter of weeks, we can regenerate bone that on its own would never grow back. And in fact, within a couple of months, we can get back about 60% of the native bone that was there before the injury. And so that's a massive amount of new bone that's growing. So this is one of our materials that we're thinking about going into clinical trials for. But I want to talk about sort of two ways in which we're also using this material going forward to make it more amenable to a wide range of populations. So there's some work of a current graduate student in my group, Aliki Koliopoulos, who's done a lot of work down at the IGB. And she's really interested in multicellular crosstalk. So what does that mean? Well, in the bone environment, I've talked to you about creating a material where you can put stem cells in it to drive new bone formation. But the reality is, in the wound site, those stem cells in our biomaterial will also be interacting with lots of other cells from the wound environment, including endothelial cells that are necessary to form the new vasculature we need to grow large tissues, osteoclasts, which you and I have in our bones and are responsible for degrading old bone, and a number of other immune cells such as macrophages. And so Aliki's been doing some work to think about how do we use our materials to understand not only how do we have a material that drives efficient bone formation from stem cells, but also allows other cells in the wound environment to take part in the regenerative process. And one of those was an observation we made that when we put stem cells in our scaffold, not only does it produce a lot of new bone-like cells, but those cells also secrete a small molecule known as osteoprotegerin. This was really exciting to us because osteoprotegerin is one of the signaling factors that inhibit the ability for osteoclasts to resorb bone. So you and me, normally without an injury, we have osteoblasts that produce new bone and osteoclasts that resorb old bone. So as you get microfractures or other features in your bone through aging or wear, the osteoclast will eat away the damaged part of the bone and osteoclast will go in and produce new bone. So what Aliki has sort of identified in this work is that not only will our scaffold induce new bone formation, but it could also prevent osteoclasts from resorbing that new bone as the healing is happening. So we did some work with a group at UCLA to really show that on its own, our scaffold really not only drives new bone formation, but inhibits osteoclasts from acting. Another question that Aliki's been involved in with another student in my group, Alec Tiffany, has been also trying to understand sex-based differences in healing. We know there's some differences in some bone pathologies, such as osteoporosis, but one of the challenges we're faced with in working with some of our clinical partners is we know within our communities, there's some patients that heal better than others. You know, some of us are super healers, some of us don't heal as well. And we're really trying to understand how do we design materials that are most supportive of a broad range of patient populations. And so Aliki and Alec did a really exciting experiment, is that we partnered with a company based in Colorado known as Essent Biologics, which has allowed us to get access to a large number of patient specimens. So we're not dealing with cell lines or stem cells from an animal, but actually adipose derived stem cells taken from a large number of patients. And we used a single one of our material variants to ask the very important question. If we have a single biomaterial that we know is really good for bone repair, do we see differences in how different patient cells behave on that? And so Aliki and Alec have screened a wide number of different factors associated with bone repair and found some really interesting observations, at least here linked to donor sex, where we find that cells from male donors were much more metabolically active in our scaffolds, meaning they were proliferating and growing faster compared to female donors, which might sound like male donors would be better. But one of the other things that was really interesting, that wasn't always the case. So this is a plot here looking at secretion of that osteoprotegerin factor that's known to inhibit osteoclast activity and was really important for our bone to functionally regrow. And here again, we see some sex-based differences where a number of female donors were producing much more osteoprotegerin. And in fact, we saw cells from female donors had higher ALP activity, which is an early marker of new bone formation, and that scaffolds seeded with cells from female donors had much more calcium and phosphate crystal mineral deposited in the scaffolds over time, suggesting that there might be some important sex-based differences in how cells from different patients would respond in our materials. And so that's an avenue we're exploring now to understand how can we create versions of our material for older patients or younger patients or patients with underlying diseases such as diabetes to support maximal bone growth. So I want to change gears a little bit, mainly so that we can talk about some other exciting avenues. I could spend a lot of time talking about some of the model system work we're doing, but one of the other challenges we have is we need to get these porous sponge materials into clinical scale defects. And so one of the other real advantages we have on our campus is we have a fantastic College of Veterinary Medicine and collaborators at the School of Animal Science, the Department of Animal Sciences, to help us test our materials in much larger models. So what you see on the left here is an image of a 200-pound Yorkshire pig on South Campus, where we're able to create clinical scale defects for humans. So these are 25 millimeter in diameter, an inch in diameter, and almost an inch, a half-inch thick defects in the mandible of these pigs we can create. And these are really clinically relevant challenges. So we're not just growing back a little bit of bone in a mouse, we're growing back half a mouse worth of bone. So these are the types of models that allow us to really show the value of our work at the clinical scale and get into clinical trials. We noticed two problems as we started to work on our materials to get into these larger defects. One of which is just the strength of the material itself. The thing that makes these scaffolds so exceptional for cell growth is that they're really porous. Cells can get into them, nutrients can get into them, oxygen can get into them, but also that means that all their pores make them relatively soft, which I'll get back to in a second. The other thing that was a really challenge is micromotion. You get an implant, you try to put it into a defect. You don't always have the luxury of being able to pre-design an implant to perfectly fit a defect. And so if you have a defect that is at all bigger than the implant you put in, your implant's going to move around a little bit. And what you end up with is this scar tissue that forms between the implant and the surrounding wound site, makes it really hard for good healing. So we're trying to think about strategies to address how to make the material stronger and how to reduce micromotion. So as a quick aside, I thought it'd be an interesting take on for a public-facing audience. But when you have a porous material, they're really cool in how they behave mechanically, like how they respond to loading. So I have an image on the right here. This is of a elastomeric a polyurethane foam used in seat cushions. It's a low-density open cell foam. Its structure looks a lot like our collagen scaffolds from a few images ago. And in fact, these foam materials are everywhere in our environment, right? We use porous materials because they're incredibly strong for their weight, but they have really unique mechanical properties that are important to understand. So as we oftentimes think about mechanical performance of materials with something known as stress strain curves, where stress is a measure of how much force you apply to a material. It's force divided by area. So you imagine applying force over an area. If you apply the same amount of force over a much smaller area, you're applying a lot more stress. If you apply the same amount of force over a much bigger area, you're applying less stress. Now, so that's, you put load on a material, and when you load it, it deforms. And so strain is a measure of deformation, where it's unitless. So what that means is the way you calculate strain is you say, how much did the material deform relative to how big it was? So if you take a piece of Café, for example, that is 10 inches long and pull it to 11 inches, you've deformed it by 1 inch relative to 10 inches. So it's 10% strain. So thinking about that, as you go to higher levels of deformation, you have higher amounts of stress that you'd have on your material. And so that's typically the way we think about mechanical performance. But in a porous material, it's added because you have all these pores. So what you see is what's known as a linear elastic regime. That's a regime where the pores are starting to deform. The fibers that define the pores start to bend. And so you can see it graphically here on the bottom using, these are honeycomb materials we made out of rubber. Initially you load it and it just sort of, everything deflects a little bit. You reach a point known as the collapse plateau, which is where all of a sudden the fibers that make up the pores start buckling and the pore collapses on itself. And you see that in this point too, where you start to see what's known as the strain band, a region where the pores start collapsing. And as soon as one pore collapses, the one next to it collapses and it cascades, you get these strain bands that form. And since everything's collapsing, what you get is you put just a little bit of load and get a huge amount of displacement. The material collapses on itself. And eventually you get to the point where all the pores are lost and you what's known as densification. So all the pores are lost and now you have the solid material pressing on itself and the slope goes back up. So what we want to do is we want to design porous materials to stay in this linear elastic regime, but be as strong as possible. So we want the slope to be as high as possible. The challenge is the mechanical performance of porous materials scales as its relative density, or how much stuff is in it. So if you have a material that is 1% dense or 99% porous, and you make it 2% dense or 98% porous, the math tells us that material that is double the density is now 4 times as strong. So we can make materials that are much stronger by adding more material into our scaffolds. The challenge is that makes them incredibly less porous, which means cells don't grow in and growth factors don't get in and nutrients don't get in. So this is not a strategy we can pursue. So we started thinking about how do we address this challenge more globally of porous materials we want to use for regenerative medicine. And so a graduate student in my group, Marley Dewey, has been really involved in using 3D printing methods. And how do you develop composite materials that will solve multiple challenges? And so a great composite we all know about in our community is engineered concrete. Like concrete is all over the place. Concrete is great in compression. not very good in tension or twisting, right? Whereas the rebar that you put into the concrete is good in tension and it's good in twisting. So by putting them together, you have a composite material that not only is better in all those modalities, but has really well-defined properties. So you know how it's going to behave. You can have a rebar infused concrete that's this size or this size, and we know it'll have scalable properties that you can depend on in your designs. So Marley was interested in developing multi-scale composite ideas for our biomaterials. We got really excited by some of the real advances in additive manufacturing over the last few years. And so an image on the left that I have is a really cool collaboration between a 3D printing company known as Carbon 3D and Adidas. And Carbon 3D has been developing really rapid printing-based methods. And so they now have a whole line of sneakers through Adidas where they print the soles of the sneakers. So the way they do it, and this is one of the Futurecraft shoes right here, is they've designed different pore architectures that have different porosities, which mean each one of these unit cells has a different strength. And then for Adidas, they made a collaboration where in order to create a shoe sole, they figured out a computational method to stitch together these different pore architectures. So they had stiffer regions and softer regions, and they could print this out as an entire sole. Super exciting, not quite what we're doing, because one of the challenges that Adidas has to deal with is that if they want a size 8 shoe versus a size 13 shoe, they have to print different soles. We wanted something slightly different. We wanted a small, a medium, or a large size scaffold that could be cut down to a defined space in the operating room and have dependable properties. So what Marley developed was an approach known using Voronoi foams. And Voronoi foams sound super fancy, but basically they're random three-dimensional structures where you take a 3D space you want to print something into, and you litter it with what's known as seed points. And the Voronoi foam is based on the number of seed points and the spacing between them. And you can print these out. These are actually printed in the IGB in one of our 3D printer systems that we could develop these foam architectures. The neat thing is, as you load them, you'll see the fiber starting to bend and buckle and the pores collapse. But these are properties we can control at the millimeter scale that are much larger than the cells in our material. But this is the scale that the surgeon would handle it on. So this is a larger reinforcing structure we're going to be penetrating into our scaffold. And so that's really what Marley had developed. So here's one of these large three-dimensional foam materials that is based on these Voronoi foams that she's printed. She then takes it and is embedding it into the slurry of our collagen suspension and then freeze drying it. And what you're left with is a material here on the bottom where it is a fiber array impregnated into our scaffold. So the scaffold is the part that drives the cells to do what we want, but isn't mechanically strong. But then we have these fiber architectures that are now embedded into the scaffold that make it strong, and also make it so that we can cut it and shape it. And the fact that these are based on these Voronoi foams means that as we cut and shape the implant, the mechanical properties of the smaller bit are the same as that of the larger bit. So we have properties that we can depend on. And one of them is what's known as compressive strength. And so these materials, now our surgeon can cut it to be slightly larger than the defect. They can squeeze it, put it into the defect, and it springs back and fills shape. So that means we get really nice conformal fitting and a strong implant that supports bone growth. So we're really excited about getting this into clinical trials over the next couple of years, where you have a material that's really exceptional for bone healing and a mechanism by which surgeons can handle it and cut it and shape it to fit all the different types of defects that they're faced with in a clinical scale. There's a lot of other work we're doing in this space. I won't really have time to talk about much of it. One I do want to point out is some work, Valencia Lewis-Charles in the group, who's really interested in some of the challenges particularly faced by diabetic patients who have notably poor healing of bone defects. And so Valencia is asking some questions about what about the diabetic environment might affect healing. And the two that she's really interested in is we know there's some differences in diabetic patients in terms of some of the metabolic signature of their cells. So the population work we're doing is really important for that. But another one is that the presence of diabetic patients' tissues actually have different levels of cross-linking due to the chemistry associated with diabetes. And so Valencia is also interested in, can she mimic some of the differences in the cross-linking of our collagen scaffolds, which affect things like mechanics and cell activity. And so we're really thinking about ways in which we can continue to expand on this work to treat a wider range of patients. I'm going to use the second half of my talk to talk about some of the other opportunities for biomaterials. And this is not work to develop materials to go into a patient, but rather how do we develop models of tissues to use outside of the body? So again, we think back to that image I showed a while ago about the tissues and organs inside of our body. And, they're really incredibly complex in terms of the tissue architectures and the like. And so you might think about, if we're starting to build models of tissues we use in the lab, how do we get towards some of that complexity? The first types of experiments people in our field have done were taking individual cells from a patient and adding different cytokines, growth factors, chemicals into liquid media, keeping it in an incubator in a petri dish, and measuring how those cells responded to those growth factors. We're really interested in how do we use some of our biomaterial tools to develop a three-dimensional matrix to encapsulate cells inside of, so that the cells are exposed to some of the native handholds, such as what are known as integrins and ligands. So, when you and I might meet pre-pandemic, we would shake hands, say hello. That's an example of how two humans might interact. And so how two cells might interact is similar. They use a mechanism through cadherence about how they would interact with each other. But cells also have a huge ability to sense the tissue environment around them. So a great example I use is, if you've ever gone to a rock climbing place to do rock climbing, right? They have all these different colored handholds that you can follow up. And that, if I've ever gone, my hands are kind of big and it's hard for me to get into the smaller grips. Maybe my grip strength isn't so good. So I preferentially go for some of the handholds that are really big and chunky and I can hold on to. My kid, who is 5 and a dynamo, she would probably just go for all the little ones and scale her way up, right? And so the idea of integrins is that they are essentially that mechanism by which how cells interact with tissues. Integrins are on the surface of a cell, and they will bind to what are known as ligands, which are on tissue. And so the different tissue architectures in our body present lots of different ligands that cells can attach onto, but they're specific. So some cells have some classes of integrins that can bind to some, handholds on the rock wall, and some cells have other classes of signals. So as we engineer materials, we engineer them to present the right ligand so that cells can attach to them. We can include growth factors that we bind into the material, similar to what we see in our tissues. But also other growth factors or chemicals can diffuse through these biomaterials to get to cells, similar to what happens in tissues. So we've been interested in how do we develop biomaterials to control cell material interactions, cell-cell contact, but also when we have multiple cells in a tissue, there's also processes that happen over time. This idea that the tissues we have are not the same throughout time. I mentioned this idea in bone where we have cells that degrade old bone and cells that produce new bone. Those processes happen all over our body. And the really exciting thing about the tissues in our body is not only are they soft, they're squishy, but they're evolving. You have multiple cells interact with each other. One cell might present a signal that diffuses to a different type of cell that's known as paracrine signaling. Cells can produce factors that diffuse back to themselves, known as autocrine signaling. And cells also produce factors to degrade the tissue around them and produce new tissue. So they're constantly remodeling the tissue. And so we're really interested, how do we build three-dimensional models to get after that? And we've developed a class of gelatin hydrogels. So this is like Jell-O. This is based off of a denatured collagen, where we use lots of different ways by which we cross-link macromers of gelatin together to form three-dimensional networks that cells can live within. And I'm going to give you an example of some of that work now. So one of the areas that we've really adapted these gelatin hydrogels for is for some challenges thinking about glioblastoma, which is the most common and lethal form of primary brain cancer. So this is a cancer that has median survival about 15 months and 3-year survival rate, 5% or so. While we've had increases in survival times due to therapeutic interventions, the overall survival rates haven't changed much in the last century. So we're not doing much better treating glioblastoma now than we were during the Great Depression. There's a number of challenges beyond that. And I'm going to try to talk to you today about how we identified a problem that we thought our tissue engineering work would be really suitable for. So we think about GBM. It is a primary cancer that forms in the brain. And it's one that grows rapidly inside the brain. And so here's a section of a patient's brain where you see this huge glioblastoma mass here on the left. What's a little less obvious is a lot of other spots that are diffuse infiltration spots of this tumor. And so a lot of cancers will kill you by metastasizing to other tissues, such as breast cancer, which primarily will metastasize to lung or brain or bone. And those are what the lethal nodules are. GBM kills you by spreading diffusely in the brain. So the way in which we treat this is that by the time a patient presents due to a neurological deficit, they usually have pretty substantial growth of a tumor mass. It turns out our brains are incredibly plastic to when they have small nodules to get around it, and you don't notice it. But typically patients will present with speech slurring or other challenges such as that. By then they have a large tumor. So what we do is our surgeon collaborators go in and they do what's known as surgical removal or debulking. So they try to take out as much tumor as possible. Then they do chemotherapy and radiotherapy on the tumor. And this is where some of the challenges come in. As I mentioned, GBM kills you by spreading diffusely through the brain. So if we compare this to breast cancer, caught early breast cancer, they try to take out the tumor. They might take out a tissue margin around the tumor, or they might do a full mastectomy. And the goal there is to remove as many cancer cells as possible. So here we have a brain cancer that spreads diffusely through the brain. So obviously we can't take out the whole brain. So you have to, at some point, put a sharp surgical margin across a diffuse cellular margin as the cells are spreading. And so what you end up with is these patients typically see rapid recurrence within a few months, and almost all of them are occurring within a centimeter or two of the resection cavity, meaning the cells that are coming back to kill these patients are the cells that escaped the outside of the tumor. We couldn't see them, we couldn't cut them out, but they're coming back with a vengeance. And so this has really motivated us. So when you look at the tumor microenvironment in the brain, inside the primary glioblastoma tumor, it's a mass of lots of different types of cells: glioblastoma cells, necrotic cells, a stem cell subcompartment that's thought to be really associated with growth and therapeutic resistance, vasculature that's growing inside of these tumors to support them, tumor-associated macrophages and microglia, immune cells from the brain. They're all in there. But the challenge is, after diagnosis, that all gets debulked, taken out. So we really identified an opportunity to develop tissue engineering platforms to study the margin of the tumor, that region that you can't cut out, the region where the tumor recurs. And we think it's a really important area for us to develop biomaterial models of the tumor. So think about these transitions between the tumor and the surrounding brain. One of them is what's known as the perivascular niche, or the vascular structures that extend from the tumor out into the surrounding brain. There's been a lot of literature and observation that glioblastoma cells, particularly the stem cells, and migrate along these vessels as a superhighway out into the surrounding brain, even to the point of popping astrocytes and other brain-associated cells off the vasculature to move along the brain. As they do that, they're exposed to a wide range of matrix proteins, so using their integrin ligand interactions, and a wide range of different cell types using these cadherin interactions than they would normally see in the surrounding brain itself. That perivascular region is rich with unique environments that they can interact with. As these cells migrate, they also form what's known as pseudopalisades, which is where these glioblastoma cells proliferate rapidly because they're right next to a blood vessel. And then they do something called a process of co-option and regression, where they actually choke off the vessel. The vessel dies, and they have now a necrotic site, or a site of hypoxia where there's not enough oxygen for the cancer cells. So a lot of these cancer cells will die, but some of them will survive this very low oxygen content, then hop to a new vessel and do it again. So these cells in these margins are going through transitions in tissue and cell and metabolic constraint that are really hard to study in an animal, but these are things we can control inside of a biomaterial. So we've also had some work that I'm not going to talk about today, but trying to develop culture platforms to grow these microglia, the immune cells that are within the brain, specifically to look at crosstalk between the immune axis in the brain and glioblastoma cells. We also have some work that's looking at how radiation damage or next generation therapies, such as antibody drug conjugates, can be deployed in the tumor microenvironment. But I'm going to talk mostly about this perivascular space today, which is really exciting. And our goal is to think about how matrix cues, cell-cell interactions, and metabolic constraint shape glioblastoma progression and drug sensitivity. So how do we do this? We've designed a class of hydrogel biomaterials to mimic the chemistry of the brain, and we started with cell lines. These are cells that were taken from a patient and immortalized. They're workhorses in our research labs because they're reproducible. They're not necessarily patient specimens anymore, but they're reproducible, so they make good calibration standards. And we developed hydrogel systems that mimic the stiffness and chemistry and ligand presentation in the brain to ask questions about how that matrix environment affects cell proliferation, how cells sense the matrix and how they remodel the matrix, and how cells invade and how they respond to different levels of oxygen. We did a lot of work with cell lines to develop a research infrastructure. We then partner with Mayo Clinic, where we can start getting patient-derived cells. So these cell populations not only have glioblastoma cells, they have stem cells, they have immune cells and the like within them. And so we can ask similar questions. How does this tissue environment not just shape invasion and proliferation, but subsets of cells? Is there an environment where the stem cells are much more likely to grow or die, or much more likely to be responsive to drug or not? And lastly, we've developed these tools to ask questions about how the matrix environment affects how drugs work. not just how a drug diffuses to the cell, but the fact that because the cell is interacting with their tissue microenvironment, they may activate a series of pathways inside the cell itself that allows the cell to become resistant to drug. So that's a kind of a terrifying concept, but it's one that the tissue environment will shape how a cell responds to drug. And if that's true, we need to develop tissue models to better understand drug screening and to better predict which drugs we should use on which patients. So I'm going to cap off this work to talk about one set of projects here related to growing a vasculature. And so this is some work of a previous student in my lab, Dr. Magno, and she had worked on developing models of this perivascular space. And so what you're seeing on the left here is a three-dimensional hydrogel that's grown with vascular cells that are stained here in red, forming blood vessel networks with glioblastoma cells all grown together to mimic this vascular region in the margin of the tumor. What I noticed was really exciting, that when we grew these structures, glioblastoma cells seemed to be very close to the vessels. And in fact, when we imaged them and measured how close they were, we found that the majority of these glioblastoma cells were all within about 30 microns of a vessel, which is incredibly close to one of these vessels and much closer than we would expect if they were just randomly distributed. So it led us to ask some questions about, is there a way by which the vessels and these glioblastoma cells would interact with one another? So I turned to some microfluidic devices. So these are miniature devices we can use in the laboratory, and the advantage is we can do a lot of experiments with not a lot of cells, which makes it more applicable to studies related to patient-derived specimens. But she did a really basic experiment that was exciting. She created a miniature brain microenvironment, a hydrogel zone in the middle of this microfluidic device, where she could add glioblastoma cells into one channel adjacent to the hydrogel, and on the opposite side, add in growth factors. So this would be conditioned media, where she would grow blood vessels in hydrogels, take all the factors those blood vessels produced, and put it into that channel on the bottom and ask the question, are the factors produced by this vasculature causing the stem cells, causing the cancer cells to migrate towards the vasculature? She found what was really exciting. The cells that weren't migrating were proliferating. The cells that were migrating were not proliferating, but more excitingly, this conditioned media from the vasculature caused the glioblastoma cells to be more likely to invade and to invade farther. And in fact, she could use a series of bioinformatics methods to identify which factors produced by the vasculature were really important for driving this glioblastoma cell invasion. So this is a therapeutic target one might look at. Something else she noticed was that when we put these cells together, not only did it seem like the vasculature was producing factors to cause glioblastoma cells to migrate towards them, we also saw this co-option regression happening. The thing I mentioned that happens in the brain where glioblastoma cells choke off vessels. We actually saw the vessels starting to fall apart in our engineered environments. And we did a bunch of genomic screens and found out that the gene expression patterns we were measuring were entirely consistent with co-option and regression. So it seemed like it wasn't a one-way street, but it was actually a coordinated conversation between glioblastoma cells and vascular cells that would shape both. And so my turn to our fantastic genomics facilities here on campus to design an experiment to figure out what was really happening in that crosstalk between vasculature and glioblastoma cells. So the experiment she did, and I'm not going to go into super amount of detail, but effectively she compared two conditions. And one was called triculture, which is where we had the vasculature and the glioblastoma cells grown together in a hydrogel. And the other was called separated culture, where we grew the hydrogel with vasculature in one environment and glioblastoma cells in the same hydrogel in a different environment that they never had the chance to speak to each other. When we did this genomic sequencing, we isolated RNA from both cultures. And with the separated culture, we mixed the RNA together after the experiment was over. So by comparing this, what we were able to do is say which genes are upregulated or downregulated specifically because the vasculature and the glioblastoma cells were grown together. And I still remember the day that Mike came back from this experiment and said, Brendan, we got to look at temozolomide. And temozolomide is the drug that we primarily use to treat glioblastoma. It's given to basically every patient that presents clinically with glioblastoma, but it has a challenge. There is a mechanism known as methyltransferase, known as MGMT, that if expressed by a patient, allows the patient to become resistant to this drug. Effectively, TMZ appends an addict to your DNA that causes the glioblastoma cell to die. But if they have this upregulated MGMT, it allows the cancer cell to take this addict off, and so the glioblastoma cell can survive, even with high doses of TMZ. So the challenge is that about 60% of patients have high levels of MGMT from the start, meaning TMZ does virtually nothing for these patients, but we don't have anything better to treat them with. And of those other 40%, a lot of them develop upregulated MGMT over time. And so overcoming MGMT mediated resistance is a really important challenge in GBM care. And the reason why this is important here is that the most highly upregulated gene of all of the genes, we looked at the thousands of them, but because glioblastoma cells were together with vasculature, was this MGMT. And so what it suggested is that the glioblastoma cells in the presence of vasculature are much more likely to become TMZ resistant. So not only are glioblastoma cells potentially using these vasculature as super highways to migrate out into the brain, but because that they're close to the vessels, they will by default be more resistant to the drug. And so we knew we had to look into this. And in fact, we pursued this in a number of avenues, one of which was this experiment here on the right, which is looking at a GR50 metric, which is how healthy the glioblastoma cells were over time. If you have just GBM cells alone in blue, as you add more and more drug, you see this value go down, meaning the cells are dying in the presence of this temozolomide. If we have the GBM cells with perivascular culture, we can put huge amounts of drug on there and the cells don't die at all. And this is in coordinated culture. We also see the effect just with conditioned media. So simply taking conditioned media from the vasculature and putting it on our glioblastoma cells means they're more likely to be resistant to drug. So this was a really exciting avenue for us. As terrifying it was an exciting avenue for us for development. So we partnered with Rex Gaskins and Paul Hergenrother here at Illinois, Rebecca Riggins at Georgetown, and Jen Sarkari up at Mayo to develop a next generation cancer tissue engineering platform, which is what we're using today. This is now a brain mimetic vasculature environment. And so Mai developed a culture system that allowed us to take microvascular endothelial cells, pericytes, and astrocytes, three of the primary cells that form the vascular networks in our brain, and grow them in these hydrogels. They form into these really complex vessels very rapidly. We see the pericytes wrapping around the vasculature, which is what we'd expect. We see astrocytes forming end feet to connect to the vasculature. They produce things such as laminin, which is the basement membrane to insulate the vasculature. And they form things which are known as tight junctions, which are markers of how mature the vessels are. And so we're able to actually form really mature brain-specific vasculature structures in our hydrogels. We then can take these and add glioblastoma cells. And so these are patient-derived GBM cells we get from Mayo Clinic. This GBM6 version is from a patient that was particularly resistant to TMZ and highly invasive. We embed spheroids or mini tumors of these patient-derived cells into our vasculature, and we can ask questions about how the vasculature affects GBM invasion and drug response. What we find is that in the presence of these mature vascular structures, GBM cells invade further and faster. Not only that, but they choose where they go. And so we started looking at, again, this distance from the vasculature. And what we found is that GBM cells are much more likely to get really close to these mature vessels, these dark blue curves, within 10 microns of a vessel. And they're much less likely to be far from a vessel. And in fact, if we look at just the stem cell subfraction of these patient-derived cells, we find the stem cell version of glioblastoma is even more likely to be close to the vessels. And so they really are forming close contact between them. We're now able to use this to look at drug response. So we have a series of new compounds produced by Paul Hergenrother's group here at the University of Illinois, which are thought to overcome this MGMT-mediated resistance. We have a perfect way in which we can test them. So some of the ongoing work supported by the NIH is using these really complex tissue engineering models to screen a wide number of new compounds before they go into clinical trials. And so this is really the power of using discovery-based science, where we think about what is the challenge that we can replicate, how can we really come up with a good model to study, and how do we use this for clinical impact. We have a lot of other projects associated with glioblastoma I don't have a lot of time for, one of which is some work of a current student, Victoria, who's really working on developing growth curves for a wide range of different TMZ variants. We have work looking at how glioblastoma cells interact with microglia and how that affects things such as migration. And lastly, we have a lot of projects where we're able to grow these cells, not only in our hydrogels, but in defined levels of oxygen content. So not just physioxia, which would be sort of the normal levels of oxygen you have in your brain, but hypoxic regions. And we find strikingly that glioblastoma stem cells in hypoxic environment accelerate degradation. So again, that's not good for the patient, but it's great for us to develop tools to understand how the matrix environment, cell contact, and metabolic constraint like hypoxia shapes invasion and drug response. So that's the end of the story I wanted to share with you today. We're really interested in how do we develop biomaterials, three-dimensional materials that can go into the body to induce regeneration, as I showed you, materials that can be used as model systems to study cancer, and with the goal of replicating features of the complexity of our tissues. And that's really some exciting work that's been going on here and at the IGB. And we're hopeful to continue to make progress and make a difference for our patient populations. So with that, I want to thank you for the opportunity. I want to end with this slide that acknowledges the fantastic team of people I have in my lab that do this amazing science, all the different funding agencies that make it possible, specifically the NIH and the NSF. We've had funding from the US Army, Mayo Clinic, AO Foundation, and the American Cancer Society to support it. All the alumni of my group that have done this amazing work, and our collaborators across campus, across the country, and across the world that make it such an exciting place to work. So I want to thank you all for the opportunity, and I'd be more than happy to answer questions if there's time. So thank you all.
Yeah, thank you so much, Dr. Harley. This was super excellent. I loved it. If anyone has any questions, feel free to unmute, or I can start reading off some that we got separately. I guess the first one that I just got sent was, what fabulous projects, all of which have great impact, do you have one that you're most excited to move forward at this moment?
Man, that's like asking who your favorite kid is, which is a terrible question to answer. I will say all of these projects are super exciting. One I didn't talk about today that I am really looking forward to its growth over the next couple of years is we have a fantastic collaboration on campus trying to develop models of the endometrium, which is the lining of the uterus. And the reason why that's a really exciting area is that women's health in general has been largely underserved and underfunded in our country. And there's a lot of really important questions related to endometrial function that are associated with lots of pathologies, including preeclampsia, endometriosis, and endometrial cancers. And so we have a group of investigators here at the IGB that are really trying to develop models of the endometriotic tissue environment. So these endometriotic lesions that form in the ovaries or form in the peritoneal cavity and can have massive issues related to pain, reproductive challenges, and the like. And so we're really excited about what it would take to develop a model of endometriosis. And so we're kind of at the stage where we were about a decade ago, where we started our glioblastoma project, where we identified, the margin of the tumor was an exciting area to try to develop tools to address questions in. We're at that stage with our endometriosis project, where we're just starting to scratch the surface of how do you develop an appropriate model of an endometriotic lesion, and how do you use that to understand some of the challenges going forward for healthcare. So that's a project that is under the radar right now, but in the next few years, I expect to really explode in terms of ways that we can make a tangible effect on that patient population as well.
Awesome, that's very exciting. Another question that came up was, where does the collagen for the implants come from, or is there like rejection issues?
That's a great question. So the collagen that we use for our implants is primarily bovine tendon derived. So it's bovine tendon collagen. We treat it to knock off some of the atelocollagen at the ends, which is where there's a lot of the immune response that you would get. This is a product that I didn't talk about it, but one of the precursors for the work we did is a product called the Integra Dermal Regeneration Template, which is it's a non-mineralized collagen sponge that is used clinically for burn patients. It's treated for hundreds of thousands of burn patients. And so when I worked as a graduate student, I worked for the inventor of that. And so I learned a lot about how to make these collagen structures there. And one of the big goals of my lab when we started it was to adapt these materials that have been developed for skin for musculoskeletal tissues. So bone, tendon, muscle, and the like. And so there's 30 years of history looking back at immune response or lack of immune response in these collagen structures if you process the collagen correctly before you make the scaffold. So we do a lot of that work in the background. The other thing I'll note, though, is that we can't say that there's no immune response, because we're taking these implants and we're putting them into an active wound site, where there used to be skin and now there's burn. Like there's a massive immune response happening there regardless. Where there used to be bone and then you had a car accident or an IED explosion, there's a huge immune response that's happening. So one of the goals we look at is, And some of the work that Aliki in my group was thinking about, are there chemical modifications we can make to the scaffold to not get rid of the immune response, but shape it in such a way that because the immune response can go a couple of ways. One, after we skin a knee, you have an immune response, and its goal is to clean out any of the bad stuff, but then also support new tissue growth. And there's sort of the transition from a more inflammatory response to a more pro-regenerative response. And so we've been doing a lot of work thinking about how do we create collagen scaffolds that minimize that massive, or that would minimize the chance of having an extended inflammatory response. You need a little of that inflammatory response to start the healing process. We just don't want it to extend into the weeks after injury. And so we don't see that. And we're thinking about ways that we can continue to tailor our chemistry so that we more efficiently push it into a regenerative stance so that we grow new tissue back faster and better.
Yeah, that's very exciting. I love learning about these processes.
No, it's fun. It's a playground of questions that we get to work in, which is one of the, just back to, one of the true pleasures of working at a university is you get to work on exciting challenges and shape what the next decade or two of research will look like. And I think that's the real privilege that we have, is that a lot of this has translational value, but we're trying to develop, what does it mean to grow a tissue? What does it mean to produce a complex tissue? How do you develop a workflow or research questions? The way in which we look at some of these experiments has massively advanced as we've tried to think about, well, we're going from one cell type, we went up 2 and 3 and 5 cells, different cells growing together. How do we study it? That bioinformatics screen I mentioned that Dr. Neo had done was incredibly difficult to figure out how even to design an experiment to look at crosstalk. And so those are things that I think are really we develop enabling technologies to let people do exciting work in the future. And that's really a real privilege to get to do.
So I think I have 2 other questions that popped up.
Sure.
One was the Boroni foam. I'm not sure if I was pronouncing that right, but what was that material made out of? Is that?
So we've made it out of a lot of different materials. There's Voronoi foam structures. Anything that's printable, you can print them out of. These Voronoi foams have been a theoretical construction that have been used to simulate the mechanics of random materials for decades back, but we can never make them. So they were entirely a theoretical construct. The advent of 3D printing meant we could start to print increasingly complex things. And so we've made them out of lots of stuff. We've used the Formlabs printer at the IGV to print out some non-biologically relevant versions we can use for mechanical testing. We've developed the ones that you saw there that are being deployed into an animal model right now are based on polycaprolactone, which is a medical polymer that has been approved by the FDA and has a slow degradation, but not a zero degradation half-life. And so that we printed with a collaborator down at Georgia Tech, although our new dean of the medical school here, we're working with him to stand up a translational tissue engineering facility that would include some new tissue engineering and 3-dimensional printing capacity so we could do it all in-house here, which would be super exciting in the next few years.
Yeah, that's amazing. And then the last one that I had was, what is the mechanism of how the TMZ treatment, how does that actually kill the glioblastoma cells?
So it ends up with a modification to the DNA in the GBM cells that leads to a cell death. If it works, that's how it does it. But this methyltransferase allows the GBM cell to basically take that adduct off and continue as if it didn't get hit with TMZ.
So that's all the ones I got in the chat. If anyone else has any questions, feel free to ask. Otherwise, thank you so much. It was great.
It's a pleasure. Thank you for the opportunity to share the work that my lab does. It was a real pleasure. So thank you.
All right.